In this video, we're going to talk about the mostly unicellular organisms, protists, archaea, and bacteria, which together make up most of the living things on Earth. So remember that all living things come from a common ancestor. So I drew it this way because one of the most important evolutionary differences is between the kingdoms of eukaryotes and prokaryotes. And you can notice that the protists are eukaryotes, while the archaea and bacteria are prokaryotes. And since karyote means nucleus and pro means before, it means that prokaryotes came before the nucleus, so they don't have one, whereas eukaryotes have a nucleus. So we're going to talk about archaea first, because they're the oldest ones. And you can remember they're the oldest ones because its name sounds like archaic, which just means really old. They were pretty much the first organisms to appear on Earth. That means they're used to really extreme environments because Earth, when it was first starting, barely had any oxygen in the atmosphere, and there were really weird temperatures and weather conditions. And they were probably also exposed to a lot of UV light because the atmosphere was still forming. So you can imagine that they're extremophiles. That means that they like extreme environments. And there are three types. There are thermophiles, organisms that like really extreme temperatures. So here I've drawn a sun to represent extreme temperatures. And there's also halophiles, which like extremely salty environments, kind of represented here by the puddle of NaCl, and methanogens, which make methane gas, which is also swamp gas or a particular type of human gas. And in order to live in these extreme environments, archaea actually have very different cell walls and cell membranes than either bacteria or protists. And for our purposes, it's not too important to know what about those membranes or walls are different. It's just enough to know that they are different. So that's archaea. Now, let's talk about protista. Protista are kind of the grab bag for unicellular organisms and some multicellular ones. It basically is any kind of eukaryote that's not a plant, a fungus, or an animal. In fact, really the only thing that they have in common is that they pretty much all live in moist or aquatic environments. And because they're part of a grab bag, they have been more easily categorized into photosynthesizing protists, which are related to plants, and these we call algae, and non-photosynthesizing protists, which are related to fungi and animals, and these are just simply called fungus-like, whereas the animal ones you might have heard of as protozoa. And this includes amoebas, while the fungal group includes slime molds. So as you can imagine, these eukaryotic protists have evolved these unique ways of getting to nutrients like photosynthesis, and they also have evolved unique movement structures such as cilia, flagella, and of course the amoebas have evolved their amoeba-like movements. And now we're going to talk about bacteria. Compared to protista or archaea, bacteria can be found in diverse environments. And what do I mean by diverse environments? Well, all around us and even inside of us. So that is anywhere from the computer you're sitting at to a nearby lake or ocean, and even inside of us, inside of our gut. And because of this, they both can help and hurt us. And so we need to get a really good understanding of bacteria. So let's focus on talking about bacteria. Here is a general bacterial structure, and we're going to go over each of these parts, which are color-coded, step by step. First, let's go over the outside structures of the bacterium. First, there is a capsule, and in some bacteria, this is a slime layer instead. And the difference between the two of these is that slime layers, in parentheses, can be washed off, whereas capsules can't. So underneath this capsule or slime layer is the cell wall. And we're not going to go into this right now, but this is different for gram-positive or gram-negative bacteria. Underneath that cell wall is the plasma membrane. And that's something you're familiar with because it's just a lipid bilayer. And now there are only two things left on the outside of the cell that we haven't talked about yet. One of them is the prokaryotic flagella. Many bacteria use flagella for movement. And the reason that I specified prokaryotic flagella is because they are different from eukaryotic flagella. And this is because it is made of the protein flagellin. So just remember that protein's name. Bacteria need this flagella to get to nutrients via chemotaxis. And chemotaxis is basically just sensing chemicals and moving towards or away from it. And you can remember that because chemo for chemical and taxi is like taxiing or moving. And the last external structure, which are all of these little yellow projections, is the fimbriae, also called opillae. And they're in parentheses because not all cells have them. Now let's talk about what's inside of a bacteria. So of course it has a cytoplasm and some ribosomes, but you should notice that there is no nucleus. It has a nucleoid area for its chromosome. So notice that that's not a nucleus. The chromosome is not membrane bound and is just sitting inside of the cytoplasm. Also, keep in mind that unlike eukaryotic organisms, bacterial chromosomes are made of circular double-stranded DNA. And some bacteria also have plasmids, drawn here in a darker blue, which are basically extra pieces of DNA. We're not going to go into too much detail now, but these plasmids give important genetic advantages to a bacteria. Now you might notice that there's one thing left that we didn't label, and those are those purple circles. They're inclusion bodies. And all they do is store stuff for the bacteria, like nutrients. And this is actually super important because prokaryotes have no membrane-bound organelles. And because they have no membrane-bound organelles like mitochondria to make ATP, they actually have to get their nutrients straight from the environment and store them in these inclusion bodies. So that brings us back to the flagella. In order to get to those nutrients, they use chemotaxis to both get to nutrients or avoid toxins. And now, let's zoom out to get a picture of what we've talked about. We talked about the differences between Protista, archaea, and bacteria, and took a good look at general bacterial structure.
Hey, welcome to another biology lesson on Make Science Easy. Today we're going to be looking at classification. In this lesson we're going to be thinking about what classification is and why it's so important. So, let's start off by looking at these shapes. What does this have to do with classification, you ask? Well, that's a pretty good question. But we can classify anything. Classification is the process of putting things into groups. So, what groups do you think we could put these things into? Have a minute to think about it. Pause the video if you need, and then when you're ready, carry on. So, the first and the simplest way we can classify things is by the very shape they're in. So we've got triangles, hexagons, circles, and squares. Each of these shapes are different. We can obviously classify them by their shape. But how else could we do it? Well, we can classify things by their colour. Obviously we have blue, we have red, we have green. All perfectly valid ways of classification. Or we can go by their size. Large shapes, small shapes. Again, perfectly valid. Finally, we can go on whether a shape has a solid colour or if it doesn't have a solid colour. All of these are perfectly good ways of classifying things. So, what is classification? It's putting things into groups, and literally anything can be put into a group. So, as we've already said, classification is the process of putting things into groups. Literally anything can be put into a group. You can classify anything you want. It's especially true of living things because it makes it easier to study. It's much easier to study a group of living things if we're studying similar things and we're looking at their similarities than if we're randomly comparing. Most classification is done on physical features, and we're going to be looking at the physical features we might classify things in later on in this lesson and in this unit. Classification also happens on many levels, so you might not choose one feature to classify things with, but you might start off with one and then add more features in as we go. For living things, the first level of classification we have is by putting them into things that are known as kingdoms. Kingdoms are large groups of organisms, and these large groups of organisms share features with each other. And we're going to learn about these 5 kingdoms in a bit. So, the 5 kingdoms, we have something called Monera, Protocista, Fungi, Plantae, also known as plants in everyday language, and Animalia. Again, we would just call them animals in everyday language. If you look at these pictures, you can see there are some similarities and some differences, but if we go deep enough, we're going to be able to see what those differences are. The next kingdom we're going to look at are the kingdom of Monera. These are single-celled organisms. This means they only have one cell in their body, never more than one. Monera are the simplest forms of life. There's nothing simpler than them. Most Monera are blue-green algae, but there are some other examples too. The most important thing about Monera is that their chromosomes are not organised into a nucleus. This means that the chromosomes are just floating free in the cytoplasm of the cell. This is very, very different to all of the other kingdoms. Our next kingdom is Protocista. Protocista are fairly similar to Monera, that they are unicellular organisms. They only have one cell in their body. The big difference between Protocista and Monera is that in Protocista, the chromosomes are found in the nucleus. This is the easiest way of telling the difference between the two. But Protocista also have other more advanced features. Some of them contain chloroplasts that help them to produce food. This is very similar to plants. Other Protocista might absorb and digest food. This is similar to animals. Also, Protocista have the ability to move. Now this is very, very different from Monera. They can either move by making the cytoplasm, the jelly within their cell, flow, or they may have hairs on the outside of their body called cilia. These can move in a wave-like action, or they might have a whip called a flagellum. Again, this can move in a whip-like action, a bit like a boat, a rowing boat perhaps, making them move. So Protocista are more advanced than Monera. The next kingdom we need to look at is the kingdom of fungi. These might look very similar to plants, but in actual fact they're very, very different. The first big difference is that they don't contain cells, they contain thread-like hyphae. Fungi are sometimes microscopic, quite small, sometimes macroscopic, quite large. We can see macroscopic things with our eyes. Another big difference between plants and fungi is that fungi do not have their genetic material in a nucleus. It's spread out throughout the cytoplasm of their hyphae. Plants produce their own food through photosynthesis. Fungi cannot produce their own food. This means they need to consume something else, and they do this by digesting dead organic matter which they actually grow on. This makes them part of a group called saprotrophs. Now, the groups we've looked at, or I should say the kingdoms we looked at so far, the Protocista and the Monera, they generally reproduce asexually. This means they make copies of themselves. Fungi are far more sophisticated. Fungi reproduce using spores. This means that there's genetic variation and there is sexual reproduction going on. Fungi are also very useful. We use them to make things like bread, to make cheese, to make alcohol, and we use them quite often. In fact, we eat things like mushrooms as well. So fungi really are a very, very useful kingdom for us. The next kingdom we're going to look at is the kingdom of plants. Most plants are multicellular. Not all of them, but most. They vary in size from being very, very small, microscopic, to absolutely huge. The largest organisms on Earth at the moment are the giant redwood trees. They are huge, huge, huge trees. One of the key features that plants have that no other kingdom has is that the cells contain a compound called cellulose. This is used for plants to make things like their cell walls. Plants also contain chloroplasts. Chloroplasts are used by the plants to produce their own food in photosynthesis. They turn carbon dioxide and water into glucose and oxygen. Some protocysts can do this, but it is mainly plants that have the ability to produce their own food. The final kingdom we're going to look at is the kingdom of animals. This is the kingdom that we belong to. Animals are generally multicellular, and the big difference between animals and plants is that animal cells do not contain cell walls. This is because animals do not contain cellulose. Also, animals do not contain chloroplasts. That means we cannot create our own food by photosynthesis. Animals need to get their energy and their food by digesting other organisms and obtaining the nutrients. When we think about classifying things, we need to make sure that everyone is using the same system. If people are using different systems, well, their classification isn't going to be the same, and this means it's not going to work. Every single scientist needs to be classifying in the same way. And the system that we all use was developed originally by a scientist called Carl Linnaeus. He worked mainly on plants when he started to classify those, but as he got on and he developed a system, he decided to work with animals as well. It's not exactly the same system that we use today. It's been refined and improved over time, but he would still recognise it. And this is the system he used. First of all, everything was put into a kingdom. These are the general features that we've already seen, our 5 kingdoms, and organisms within a kingdom are loosely related. He then puts things into a phylum. Things in a phylum are slightly more closely related than in a kingdom, into a class, a subclass, an order, a family, a genus, and finally down to a species. Things within the same species are incredibly closely related. So as we go through Linnaeus' system, every level we go, things are more closely related. Linnaeus also developed another system, a naming system, and it's called the binomial naming system, and this is because every species has 2 names. This is their genus and their species. So every single species has 2 names, and we still use the system today, and these binomial names are usually. So, binomial naming is the naming system that we use today. Species are named by their genus and their species. Regardless of which language is being spoken, the species name remains the same because it's in Latin. Now, when we write a species name, it needs to be written in italics. Now, this is pretty easy if we're using a computer. If we're handwriting, it's not always so easy, so if you're writing a species name, you should underline it. Now, the first letter of the genus must always be a capital letter, but the first letter of the species should be lower letter. So remember, genus, species. First letter of the genus, capital. First letter of the species, lowercase. So let's think about humans as an example. We are Homo sapiens. Homo is our genus, has a capital H. Sapiens is our species, has a lowercase s. Our binomial name is Homo sapiens. We could also look at some close relatives of ours, Neanderthals. Now these are also part of the genus Homo, which means they are closely related to us. So they form Homo neanderthalensis. Homo, their genus, Neanderthal, their species. Capital, lowercase. So, in summary, scientists classify living things to make them easier to study. The first level of all classification is to put organisms into one of the 5 kingdoms: Monera, Protocysta, Fungi, Plantae, Animalia. We then classify all living things a little bit further. All living things are given a binomial species name. For example, Pan troglodytes, which is a chimpanzee by the way. Classification, however, is not always easy, and it's sometimes very challenging to classify an organism completely. Now, I hope you've learned a lot in this lesson. In the rest of this unit, we're going to do some more classification. Remember Make sure you use the resources provided with this lesson. Make sure you have a go at the multiple choice quiz. Make sure you've understood things. If you haven't, give everything another go. Until next time, hope I made science easy for you. Goodbye.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about fungi. Fungi is a pretty diverse group. It's things that you're familiar with, like a mushroom, but it's also things that are much smaller than that, like a yeast, which are going to be single-cell fungi. And they're also going to form sometimes symbiotic relationships. So lichen, for example, is a symbiotic relationship between a fungi and then an algae. And so not only do they break down and decompose material, but they're also going to serve another role, working symbiotically with both algae and then plants to allow them to function better. And so basically, if we look at their phylogeny, so this is the eukaryotes. So we're going to talk about things now that have a nucleus and have organelles. And you can find here that here's our fungi. In other words, when we try to classify all eukaryotes, we're going to put them right here. If you look at animals, animals are going to be right here. And so what does this mean? Well, we're going to share more recent common ancestry with fungi than we are with plants, which are going to be way back here, because we'd put green plants way over on this side. And so we probably have more in common with a fungi, especially the way we live our life, than we do with green plants. But we're not directly related to them. This is more into the phylogeny of the actual fungi. And again, we get this from the Tree of Life project. And so you can see that we have this break off like this. And there are monophyletic groups. In other words, there's going to be some groups that you should know, because we know that these things all share common ancestry and they're one group. And so there are going to be 5 of those: Chytridiomycota, Zygomycota, Glomerulomycota, Ascomycota, and then Basidiomycota. And so these are ones that you do need to know because they're monophyletic and they're important when we're studying biology. But before we get there, let's get to the characteristics of fungi. Well, they're eukaryotic, so they've got nuclei and organelles. They're heterotrophs. They originally were classified as plants, and you can see why. It almost looks like a plant, and they don't move, they don't run around like animals, and they almost have these root-like structures that look like plant roots. And so we used to classify them as plants, but they're not. They eat material, and they're going to digest material. I'll show you some cool examples of that in just a second. They have cell walls, so that separates them from us. Also the fact that they don't move. And those cell walls are made of a polysaccharide called chitin. The exoskeleton of insects are made from that similar structure. Most of the inside of the structure is actually going to be filamentous. In other words, it's made of filaments. And so this would be the filaments in a mushroom. You can see these thin little filaments. You can see the cell wall going around the outside of it, but they're not going to have any true tissues. In other words, they're not going to have, for example, muscle tissue or nervous tissue. It's just going to be a group of these hyphae, they call these. That's what these filaments are called. Over and over and over again. And so even if we were to look in here to these fruiting bodies of these mushrooms, we'd find that they're hyphae just packed together really, really tightly. Or if we were to look at the roots of them, those are going to be hyphae absorbing nutrients. And so those are some of the characteristics shared by all fungi. They're all heterotrophic. And so if you see one growing out of this tree or this rotting tree, they're digesting the material inside there and they're eating. And you can see here that this mold, which mold is kind of a term that just means a fast-growing fungi. You can see this mold is breaking down this fruit. And if you've ever had athlete's foot, you've been infected by a fungus as well. And so this right here is a fungus growing in between the toes, and its hyphae will actually grow into the tissues of the person's foot. It secretes an enzyme, and then it digests that material inside. And it's really painful because it's starting to digest the nerves on the outside of the foot. And one of my favorite fungi of all is this Anthrobotrys. Basically what they do is you can see the hyphae that they're growing here, but they'll make these tiny little rings. And then when you have a worm like this is called a nematode worm. Basically, if the nematode worm starts to swim through this ring, it will cinch tight on the nematode worm. It'll grab a hold of it, and then it's going to digest it from the outside in. And so this is an animal-eating fungi. And so what could be much cooler than that? How do they reproduce? Well, they do reproduce a lot of the time asexually. And so when you have mushrooms just continuing to grow out and out and out and out, and they can grow relatively quick, that's going to be asexual, but then they can also have a sexual phase as well. And so the spores that come out of a puffball like this are usually going to be asexual spores. They can spread to an area where there's a little bit of water, some food, and they can start growing. But there's also sexual portions, so these were formed through a sexual process. If we look at this mushroom right here, basically what you get this would be a basidiomycete. Basically, you're going to have spores that fall down. You get the growth of a new mushroom, but then we can have sexual reproduction producing more spores. And so there's a variety of ways they reproduce. Like I mentioned at the beginning, there are 5 important phyla that you have to know when it comes to studying fungi. And I classified them from A to Z. This is just a quick way that I remember it. So we've got A, B, C, and we've got Z over here. So we go from A to Z, A, B, C, and then we got G in the middle, and the G can stand for golly, aren't those fungi cool? Because these ones are going to be really cool in just a second. And so let's go from A to Z. So the first ones are the Ascomycota. Ascomycota, the asco or the ascus, that's what ascus means cup. So ascus means cup fungi or sac fungi. And you can see right here is this is that sac, and then the spores are going to form on the inside of that. And that's why we call it the Ascomycota. Why are these important? Well, this is my favorite fungi of all. This is the morels. We like to go hunting them in the spring. They taste delicious, but they're really hard to find. You're looking right here at the sexual portion of that, but you can see these sacs on the inside or the sac right here. But Ascomycota are also important. So penicillin is a fungus or an ascomycote that is going to make a lot of the antibiotics that we have. And so they're important for that. If we look at Basidiomycota is the next one, so that's the B. Basidium comes from the word basidium, which means club. And you can see that on the underside of a mushroom, they're going to have these little gills. But if we zoom in closer, we're going to find these club-like structures. And each of those club-like structures is going to form spores. We'll have 4 spores that are found on this basidium, and that's where the name Basidiomycote comes from. It's club fungi. And so what are the big ones? These are going to be the mushrooms that we think of when we think of fungi. Those are going to be Basidiomycota. If we look at these two together, sometimes we would say these two share common ancestry, Ascomycota and Basidiomycota, and we sometimes call these the top fungi. What's next? Next is the C, so that's going to be Chytridiomycota, and these are going to be sometimes referred to as the chytrids. These used to be classified as protists, and the reason why is that they have a flagellated stage where they'll actually swim around. We now know that they're true fungi. You've read about these in the news because we've seen on the planet huge increases in deaths of frogs, and a lot of those are related to chytrid fungi that are infecting the frogs. All right, let's go to the G then. So the G stands for the Glomerulomycota. Glomerulomycota are very important because what they'll do is they form a symbiotic relationship with plants, or a plant symbiosis. So what we have are these mycorrhizal relationships. And so this right here is the root of a plant, but you can see the fungi growing inside it. And almost all plants, in other words, every plant that we look for has these relationships between the Glomerulomycota and their roots. What does that give them? Well, that gives them increased surface area, and it's allowed them to take in more water, and then they give these fungi a place to live. And so it's a great relationship for both the fungi and for the plant. And then the last one's going to be the Zygomycota. Zygomycota, its name comes from the zygosporangia, which is going to be this resistant structure where the spores are actually formed in. It'll rupture, and then we get this a hiss as those spores are spread out. If you look at that, a bunch of them together look like that, and a whole bunch of them together are going to look like this. So this is a bread mold, zygosporangia, that you're familiar with. And so basically what happens if you leave some bread out, one of these spores is going to land on it, and we're going to get the growth of this fungus. What's the fungus eating? It's eating the bread, because that's what they do. They're heterotrophs. And so can you remember those 5 important phyla of fungi? Ascomycota, Basidiomycota, Chytridiomycota, Glomerulomycota, and Zygomycota. And they're all just different types of fungi, and fungi serve a huge role on our planet. They break down material when it's dead and dying. They recycle those nutrients, but they also form these symbiotic relationships with plants and algae. And I hope that all is helpful.
When plants appeared on the land for the very first time, these plants were extremely tiny, like moss and algae. And for a few million years, there were no plants on our planet which could grow taller than few inches. But why? Well, turns out growing tall has some challenges. However, with time, plants slowly started evolving, and eventually they were able to overcome those challenges. And as you know, today we can have tall plants, plants which can grow into trees, trees which can grow hundreds of meters tall. And so the question we're going to try and answer in this video is, what are the challenges that the early plants faced because of which they couldn't grow tall? And how did they overcome them? So, the biggest challenge that these early plants faced was a lack of a transport system. Okay, what's a transport system and why do they need them? Well, let's take an example and understand this. For example, consider my houseplant. In order for this plant to stay alive, all the parts of it require food and water and minerals so that they can grow. But where are they available? Now, you might know that most of the water and the minerals are available in the soil. So the soil contains water and it has minerals in it. Minerals. And the plants have roots which can absorb these water and minerals. So great. But what about the rest of the part of the plants, like the leaves? How do they get that water? Well, can you see the plants need to transport that water from the roots all the way to the leaves and all the other parts. So can you see that a transport system is needed to transport this water and its dissolved minerals up, right? Similarly, think about food. How do plants get their food? You might know that they photosynthesize. They use the energy of the sun to create their own food. And most of this photosynthesis happens in the leaves. Okay? So let's say some of the food is synthesized in these leaves. Consider these are food. Okay, these are basically sugars. Let's just call them as food as of now. Now, what about the parts of the plants that cannot photosynthesize? For example, roots can't photosynthesize because they're under the ground. They're not exposed to sunlight. There may be certain growing parts of the plant which might require this food because they cannot photosynthesize. Maybe there are some leaves somewhere which are in absolute shade and because of which they are not able to photosynthesize. So again, what to do? We need a transport system. The food needs to get transported to the different parts. So again, the food needs to get transported down, maybe to the roots sometimes. It might have to get transported up to some other parts. So can you see a transport system is necessary without which these plants cannot survive? And the earlier plants didn't have a transport system and therefore they were very tiny. The thing is, if you are very tiny, then this transportation happens just by diffusion. Diffusion is a process in which stuff moves from a region of high concentration to lower concentration. So if it's very tiny, these food particles, these sugars can easily move into the different parts of the plant. They can easily diffuse into it. Similarly, the water can easily diffuse to all the different parts of the plant. But as the plants get taller, then diffusion becomes super slow and it almost becomes impossible to transport it just by diffusion. This is where specialized transport system is needed. And so eventually plants evolved these transport system. Today they are called as vascular tissues. So the transport system evolved are called vascular tissues. And the word vascular means vessels. Vessels. I like to think of them as pipes, so you can think of them as pipes. So piping system through which they can move the food and water all around their body. And what do these vascular tissues look like? Well, just like the name suggests, they look like pipes. So pipes which start from the roots. So let me show you the roots. And so pipes that start from the roots and go all the way to each and every leaves. I've not shown it for all the leaves, but it goes to each and every leaves. But wait, you may be thinking, how can we use one pipe to transport both food and water? Won't they get mixed up? Well, to make sure they don't get mixed up, we have two separate piping systems, one for water and one for food. And we give them names. So the piping system that we use for water is called xylem. So xylem transports water and the dissolved minerals. Okay, when we say water, it already has dissolved minerals. And for food, we have another tissue called phloem. Oops, it's phloem. And this one transports food, mostly sugars, but it can also transport other things like hormones, amino acids, and other stuff, but mostly food. And the way I like to remember this, I remember watching this in some YouTube video, I like to remember this as F for food and F for phloem. So food, phloem, F. And another reason why they have to be separate is because if you look at water, the water transport is only one direction, up. Can you see why? Because all the water and minerals are present in the roots, right? So we just have to transport it up to all the leaves. But the food transport has to be in both directions. Sometimes you would want the food to go up, maybe to the growing areas of the plants. Sometimes you want the food to go down in the roots. That's where the storage organs are, right? So you see, food needs to be transported in both directions. And therefore, xylem is only one-way transport up. And phloem, well, that is a two-way transport. So it makes perfect sense to have two separate vascular tissues altogether. And so this white line I have drawn, don't think of this as a single pipe, but imagine it's a bundle of a lot of xylem pipes and a lot of phloem pipes together. In fact, let me show you a little better. If I were to zoom in onto this, and if we could see inside, these are the xylem and the phloem bundles. Again, each one has a lot of xylem and phloem tissues inside. And if you could see from the top, if you could have a microscopic view of this, right, then it would look somewhat like this. This is what you would see in a microscope, okay? And what you're seeing over here, these are the vascular tissues. Again, let me zoom in even further. So if you zoom in one of these parts, you can see a lot of round things over here. These are the xylem. Let me just mark them. These round things that you're seeing over here, these are the xylem tissues. They are the pipes that are coming out of the screen. Okay, think of it that way. They are the one that transport water, these over here. And then you can see pipes over here as well. You can see small round things over here as well. They are much thinner than xylem. These are the phloem. Okay, in between you have other cells, but these are the phloem. And so you can see a lot of xylem and a lot of phloem are bundled together along with some other supporting cells as well. And that bundle is what I've drawn over here. So the single thing that I've drawn over here is both lots of xylems and lots of phloem together. And these pipes, they start from the roots and they end all the way into the leaves. So let me show you a zoomed in version of the leaf. Right? You may have seen a leaf like this. The veins that you see in the leaves are actually the xylem and the phloem. These are the vascular tissues. And then you can see they branch off into becoming smaller and smaller. Let me zoom in even further. And you can see them branching off like a network of roads connecting different parts of the city, right? This way they make sure a maximum number of cells are able to get the food and water. That's how the transport system in plants work. Isn't that amazing? And now that they have developed the vascular tissues, nothing can stop them from growing tall, growing into trees to grow hundreds of meters tall, because of which we have forests and jungles and everything. But finally, you might have another question. How do these transport systems even work? I mean, in tall trees, how are they able to get that water from the ground all the way to the top? Is there some kind of a pump? And I guess that's where things get even more interesting. No, they don't have a pump, yet they use some clever mechanism to get their job done. And we'll look at these mechanisms in great detail in the future videos, okay? But one of the things about not having a pump is that this whole system becomes very slow. So plants, because they don't have a pump, they have slow transport system. But you know what? That's fine for them. The plants are saying, well, it's okay. We don't have a rush. We don't have to go anywhere. They have pretty low energy demands, and so they can easily survive with a very slow transport system. They don't need a pump. Unlike animals who have very high energy demands, and animals do need a pump. That's why we have a heart. Heart is our pump. But plants and trees, they don't need that. And as I said, we will look at how these things work without a pump in the future videos. Anyways, that's it for this video. So let's quickly summarize. What did we learn in the video? We saw that plants have developed a piping system to transport food and water, which we call the vascular tissues. We have two different ones. One to transport water, unidirectional transport called xylem, and one to transport food in both directions, bidirectional, called phloem. And the way I like to remember this is ph for food, ph for phloem. And since they don't have a heart, their transport system is pretty slow, but that's fine because their energy demands are pretty low, as they don't have to go anywhere. They're pretty much still. And I would like to end by asking you to wonder about one thing. Can you imagine how different our world would be without vascular tissues?
Hello everyone, in this video we're going to talk about animals. So the first part we want to mention is characteristics that all animals have, which are they're all multicellular, they are heterotrophic, meaning that they cannot make their own food, they have to eat food in order to survive. They're all eukaryotic, meaning they have, all the things that eukaryotes have. We have a cell membranes, membrane enclosed organelles, we have a nucleus and all that. And we do not have cell walls as animals. And when we think about animals, sometimes we get a little confused because animals often have hair or feather, and we consider those as animals. But fish and spiders and scallop and jellyfish, those are actually all animals because they don't belong to plants or protists or fungi. So the only category left is animals. So when we think about animals, there are two different categories. There's the vertebrates and there's the invertebrates. The invertebrates do not have backbones or a vertebral column. You can just remember the backbone part. But the vertebrates do. And you can also call vertebrates chordates. So every chordate have 4 different characteristics, at least at one point during their development. So it doesn't mean that the adult form, the born version of this animal needs to have all 4 characteristics at the same time, but these 4 characteristics existed, possibly during the embryonic development or even after they're born. So it varies depending on what kind of animal we're talking about. But these 4 characteristics are a dorsal hollow nerve core. So dorsal means the back of you, ventral means the front, anterior is the head, posterior is the feet. So you need to know those 4 directions of the body plan as well. A nodal cord, which also kind of turns into the nerve cord, the spinal cord area, and then a tail that extends beyond the anus. So right now we don't have a tail, but at one point during our development as an embryo there was a tail, and then it goes away. And then the last one is the pharyngeal pouches, which are these structures on this picture, and they supposed to turn into gills because all of us land animals evolved from marine mammals. So fish is our ancestor, and they had these pharyngeal pouches that turn into gills. They no longer turn into gills for us, but during our embryonic development, there are still structures like that that can be seen. Pharyngeal pouches, yeah, that's what we just said. So the gills are used for gas exchange, meaning gas exchange, oxygen coming in, carbon dioxide going out kind of situation. Chordates with backbones are called vertebrates. So the next one that we want to talk about are several essential functions that animals must perform to survive. So the first one is to maintain homeostasis, the balance of something, right? It doesn't mean it has to be the same all the time, but there needs to be a balance of some sort. So you need a balance of your body temperature, you need a homeostasis of your blood sugar, the homeostasis of the amount of water in your body, the homeostasis of the amount of waste in your body, oxygen, carbon dioxide, all of the above, okay? You also need to gather and respond to information because that's what allows you to survive, right? You need to interact with the world around you, whether it's the food that you're trying to get, whether it's within the population and other individuals that you need to interact with. So you need to gather the information and then respond to it. There's also obtaining and distributing oxygen and nutrients, right? Oxygen is really important for us because every single cell needs to do cell respiration in order for you to survive. And then the nutrient part, in order to do the cell respiration, you also need the nutrients. So all of your cells need that nutrients to produce energy, ATP, to actually power all the cellular activities. The next one is collecting and eliminating CO2 and other waste. As all those chemical reactions are going on in every single one of your cells, and when all your tissues are coordinating together, this entire process is going to bring out some waste, including CO2. And then there's also other solid waste that need to get rid of, that we need to get rid of. And then there's also reproduction that allows the population to continue to survive. So these are the 5 factors, 5 functions that every single animal needs to perform to survive. The next one is what are some body features of an animal body plan. So when you look at an animal, there are some features of it that we all share. So the first one, well, that most of it, yeah, that we all share. So the first one is levels of organization, right? When we think about the levels of organization, we think from small to big. We have cells, tissues, organ system, and the organism. So every single one of those lays out and put together to form an entire organism. So those different layers of tissues, for example, under your skin, you have all these different layers of fat and muscle and connective tissue. All those different layers of organization allows our body to work together. And then there's also a body symmetry. All the animals have a body symmetry of some sort, whether there is no symmetry, meaning that you can split the sponge in any possible direction and no 2 halves are going to be exactly the same. Radial symmetry means it's kind of a, it's like growing outward. It's like a firework kind of situation where you have this radial extension from the middle to all the ends, and you can basically, split this organism into any 2 halves, and the 2 halves are going to match with each other. So as you can see, if we cut right here, not to say you actually cut it, but if you can imagine cutting this organism right here, the 2 sides are going to be the same. You cut this side, they're going to be the same on the 2 sides as well. And then there's the one called bilateral symmetry, which is what we are. Bilateral means there's only 2 sides. For a human, we split in half and our 2 sides match almost perfectly, right? Some of our organs obviously don't match perfectly on both sides, but overall our body plan can be split into 2 halves, and they are mirror images of each other. So if you look at most insects, they're also bilateral. They also have a bilateral symmetry of a body plan. The next one is differentiation of germ layers. So when we look at a fertilized egg, which is also called a zygote, that one initial cell that we all came from, that one cell, what it would do is that it will go through mitosis and turn into 2 cells, and then 4 cells, and then 8 cells, and then eventually turn into a ball of cells. So this ball of cells is not as simple as it looks. It's not just a ball. It's different from, I don't know, it's different from an actual ball like this, for example, where it's just, all the sides are the same. When you look at it, they do appear to be the same. They're just cells, right, in a ball shape. However, they're actually different layers of this ball of cells that turn into different part of your body. So this is all pre-planned out even when you're only a few weeks old, maybe even less than that. A few days old, this little ball of cell will already give you a body plan so that you don't turn into just a giant blob of an organism, which is useless, right? We have arms and legs and head, and this, different parts of our body allows us to actually function. If you're just a ball, it's not going to be very useful, right? So there are different layers. So the first layer that we want to talk about is the innermost germ layer. Germ layer means this ball of cell. The germ is like not the actual bacterial germ. These are the embryo germ. Okay, so the innermost layer will develop into the lining of the digestive tract and the respiratory system. So if you think about your body, right, going from right here, you have the digestive tract and the respiratory system, and that's on the inside. And then we have the mesoderm, which is the kind of in-between layer where you have the circulatory system, the muscles, reproductive, and excretory organs. So excretory are your lung, for example. Oh no, wait, not. The lung is the endoderm. So your liver, for example, your your small intestine, large intestine. Well, the large intestine, I meant. The small intestine is not. The small intestine is digestive. Digestive is right here. Okay. So the excretory organs, the kidneys, for example, right? The kidneys and livers, those are the mesoderm. They're the in-between part. Your bloodstream, for example, that's your circulatory system. That's also a mesoderm. And then the ectoderm is the outermost layer where you have, if you just look at a person outside, you have the skin, you have the nerves right under the skin that allow you to feel things, right? Those are all the ectoderm. So this entire plan of endoderm, mesoderm, and ectoderm is already preset out even when you're just a ball of cells like this, okay? So that's a look of the ball of cells. And then there's also, inside your body, you have a formation of a body cavity for mammals specifically. So for humans, we have this kind of an empty space near your tummy, but it's not really empty. It's filled with fluid, and you have organs, having tissues allowing those organs to attach to the other parts of your body so they don't move around and get all messed up. So this body cavity, what it means is that there's this fluid space in between the digestive tract and the body wall. So your stomach is kind of suspended in this fluid-filled space. And then when we look at animals, we can also differentiate the different types of animals by looking at something called blastopore and what it turns into. So what happens is you have a zygote, that's your initial fertilized egg is called a zygote. And then when it divides quite a few times, you will have a little ball of cell, and this is called a blastula. So what happens is this little ball of cell will start caving in on one end of the ball, and then this caving in will either become the mouth or the anus of the organism. If this little caving in part is called the blastopore. So if the blastopore becomes the mouth, that will make you most likely an invertebrate, which is also called protostomes. So protostomes is for any organism that has blastopores becoming the mouth, and those are usually invertebrates. And then for us, we are deuterostomes, the initial caving in, which is determined like way early, 1 or 2 weeks of our life, that will eventually becomes the anus, just like this picture right here. And then the next one is all the organisms have segmentation. So segmentation is kind of different from limb formation. These arms, obviously, we have segments of our arms. This is kind of similar to this. But the segmentation, we want to specifically talk about insects that have different segments of body parts. They don't have bones, but they have they have the segments of the exoskeleton, which allow them to have different compartments of their body. And then there's also cephalization, which is the growing of a head, which doesn't apply to all the organisms or even all the animals, but a lot of the animals do have a head. So cephalization is a process of obtaining a head during development. And then there's limb formation, which happens to some animals, but not all animals. So for example, spiders, insects, they don't have limbs. They do have exoskeleton, legs. We can call them legs, but they're different from our legs, right? So we're talking about actual bony limbs, legs, flippers, wings. Those all came about from bones, and they're all kind of a homologous structure between all of us. The structures of our arm and hand is kind of similar to the flippers of fish and wings of birds. So we have all these features of animal body plans. As long as you understand all the things that I talk about, you should be good. And then lastly, we want to talk about a cladogram of animals. A cladogram is kind of a family tree of evolutionary relationship. Which organism is more closely related to which of the other organisms and which are not as closely related. And then in order to make a cladogram, we want to look at the adult body plans, right? So you can compare a human versus a chimpanzee and see a lot of just similarities just by looking at us, right? And you can also look for patterns of the embryological development. So during our embryonic stage, there will be even more similarities compared to the adult form. And obviously, you can look at DNA and protein and all that patterns, and then you form this evolutionary tree. Who's more related to who? So when you look at this picture right here, this is a pretty brief cladogram of animals. What you see right here is these are all invertebrates, and then right here we have the vertebrates. And what you see right here is there's a difference between a clade and not a clade. A clade means you have a common ancestor in all the descendants. So for example, if you look at this part and then both of these organisms, this is called a clade. If you have this part and all three of these organisms, this is also called a clade. But if you were to look at all of these, this is not a clade because they all trace back to this particular common ancestor, but not all the descendants are involved. These are not involved in this process. I know this might be a little confusing. We'll talk about it more in class on Friday. So what we do need to know is that for invertebrates, they're not a clade because not all the descendants are in the group. But vertebrates are a clade. You have the common ancestor, and all the descendants are all in the vertebrates group. Okay, that's all for this one.